There's lots of problems that can occur when there's problems with the myelin, and one of the most common is multiple sclerosis, abbreviated as MS. And this occurs when there are myelin scleroses where they hardened. So essentially, an axon that had saltatory conduction goes to having continuous conduction. So MS is an autoimmune disease, which means the immune system attacks itself. It affects females more than males, more common in Caucasians. And problems could include visual disturbances because it affects the optic nerve, weakness in muscles, loss of muscular control, or things like urinary incontinence because there's a demyelinating process that is occurring at a lot of these nerves that go to these different regions of the body. So the nerve fibers are severed. The myelin sheaths in the central nervous system become non functional due to scleroses. So short circuiting of nerve impulses occurs. And some treatments include a medicine called methylprednisolone and beta interferons. So MS is a very ugly disease. Next thing we're going to talk about are synapses. And a synapse is like the neuromuscular junction that you learned about in chapter 9. It's where two different nerves are going to meet. So it's a junction that mediates information transfer from one neuron to either another neuron or to an effector cell like a muscle or a gland. And this example, again, was what you learned about in chapter 9 called the neuromuscular junction. So with a synapse, we basically have two neurons that are communicating. We have a presynaptic neuron before the synapse and a postsynaptic neuron, which is receiving the signal. An analogy for this would be if you think about the pitcher that's at the mound, the pitcher is going to be throwing a ball to the catcher. The pitcher would represent the presynaptic neuron. The catcher would represent the postsynaptic neuron, and the ball would be the neurotransmitter. So when we look a little more closely at this diagram, we see three different types of synapses. Notice there is a dendrite, a cell body, and the axon, the three parts of the neuron. This region right here is called the trigger zone, or also called the axon hillock, because there's a high concentration of sodium channels. So depolarization happens and an action potential begins there. So the action potential will leap from node of Ranvier to node of Ranvier. So the three different synapses that we have are called axosomatic, exodendritic, and exoaxonic. Notice that the beginning of each of the names starts with axo. So the presynaptic neuron or the pitcher in this example would always be the axon at the axon terminal. The second part of the word is always going to represent the postsynaptic neuron or where the catcher would be at home plate. So here, the soma, the cell body, is where the receptors are. In this example, the dendrite is where the receptors are. And in this example, the axon is where the receptors are. And remember that in these situations, there's always a graded potential, which occurs at one of these spots, and those are sub-threshold potentials. So we have two different synapses. We have one type called an electrical synapse, and these electrical synapses are less common than chemical synapses, which we're going to talk about next. This correspond to the gap junctions. which you learned about earlier in the semester. There's a protein channel called a connexon that permits ions to flow from one neuron to the next. And they're found in the brain, and they're abundant in places like embryonic tissue. But some are eventually replaced by synapses. These are important in things like arousal for sleep and mental attention. So again, they're the most rare. The much more common ones are the chemical synapses, and they are specialized for the release and reception of neurotransmitters. So as we discussed in a previous slide, there's two parts to a synapse. There's the axon terminal, which is the presynaptic neuron, which releases the synaptic vesicle, and the receptor region, which is on the postsynaptic neuron. So remember in chapter 9, you learned that the end of the axon is an axon terminal. There's synaptic vesicles in the terminal that have neurotransmitters. Then there's going to be a region of the postsynaptic neuron, which could be the soma, the cell body, could be the dendrite, or it could be a part of the axon. But either way, it's going to have a chemical or a ligand gate, not the voltage gated channel that's in the action potential or in the axon. So this is what those chemical synapses look. This is a much prettier picture than what I drew. So the nerve impulse reaches the axon terminal of the presynaptic neuron. Voltage-gated calcium channels open. Exocytosis occurs. So those vesicles migrate to the end of the axon terminal, and the neurotransmitter is released by exocytosis. The neurotransmitter crosses the synaptic cleft, binds to a receptor on the postsynaptic neuron, and then the postsynaptic membrane permeability changes due to ion channels, so it could be excitatory or inhibitory. And those two types could either be an EPSP or an IPSP. Remember, EPSP stands for excitatory postsynaptic potential, and IPSP stands for inhibitory postsynaptic potential. So if we zoom in here a little bit more, we can see in the presynaptic neuron, the action potential is jumping down via saltatory conduction because of the myelin, and it arrives at the axon terminal. The next step is there's voltage channels that open, and calcium goes from the extracellular fluid to the intracellular fluid of the neuron. That is the signal that causes the synaptic vesicles to then move to the end of the axon terminal. So they can then release the neurotransmitter via exocytosis. Neurotransmitter diffuses across the synaptic cleft, which we see here, binding to a chemical or ligand-gated channel crossing the synaptic cleft. Then, once the neurotransmitter binds, there's ways that it can be removed because we don't want a neurotransmitter constantly bind or to constantly stay bound to the postsynaptic membrane. So one possibility is that there is reuptake of the neurotransmitter by the astrocyte, or there could be degradation of the neurotransmitter by enzymes. And the one that you learned about in chapter 9 was acetylcholinesterase. Or finally, there could be diffusion of the neurotransmitter away from the synaptic cleft. And you can see a couple Examples of this shown here. Because of the neurotransmitter binding, we have graded potential because an ion moves. This would be reuptake of the neurotransmitter. This is enzyme degradation, and this is diffusion away from the synapse.